So good morning. Just like Jean-Pierre, I would like to start by thanking Philippe for having me in this meeting of old friends. This is really nice to see you all again. Some of you I actually see every day, but some I don't see very often. So it's really great, and I'd like to thank Philippe again for inviting me. Okay, so what I'm going to tell you about is the Ameropoetic System. We have been interested since a long time. It was actually in the lab of Philippe that we did some of the main observations that I'm going to discuss today with you. When I was there, also like Jean-Pierre, maybe 20 years ago or so, I didn't really count, and I don't want to know how many years ago it was. So this is the hierarchical view of the Hematopoietic system, how it happens in the bone marrow. It starts with this hematopoietic stem cell, which is, as you all know, a cell that has self-renewal and multipotent capacity to differentiate. And it is actually this system that gave rise to the main paradigms that are today used in stem cell biology in general. Now all of this is happening in the adult in the bone marrow. And we think that the reason why this whole development occurs and how this pool is maintained by probably asymmetric cell division and differentiates to give rise to the adult cell types is basically because it interacts directly with another cell type, which is most probably derived from skeletal stem cells, which constitute the progenitors of osteoblasts, chondrocytes, and adipocytes. And what I call here myelosupportive stromal cells, I think we really don't know exactly what those guys are. I think those cells definitely make part of what we call the niche of the hematopoiesis in the bone marrow, probably endothelial cells as well, but we don't really understand how that happens. We don't really understand what signals this cell either to self-renewal or to differentiate and, lose self-renewal potential. Anyway, what we have been interested along the time is in the development of this system, and this is a very simple scheme of how the hematopoiesis develops in the mouse embryo. We start by the yolk sac, which is where the first hematopoietic cells from embryonic origin are detected in the yolk sac blood islands. Then you have, then the heart of the embryo starts to beat, and blood starts to circulate from those blood islands through the body of the embryo. And then there are some major things happening. One of them is that fetal liver, which is the embryonic hematopoietic organ, gets colonized. And the fetal liver is always colonized by exogenously derived cells. And so one of the major donors of early multipotent progenitors are even stem cells. I'll tell you a little bit more about that later, are originated in this region, which is called the aorta gonad mesonephros, or also called AGM short. So we think the fetal liver is colonized by these cells. The hematopoietic stem cells actually expand and differentiate in the fetal liver, unlike in the bone marrow, where they only differentiate and self-renewal, and then they colonize the thymus, the spleen, and then later the bone marrow. And it has actually been directly shown that the cells that are produced at this time point here in the mouse embryo are actually the cells that colonize the fetal liver and the same cells that colonize the bone marrow, okay? So this is what happens. So this is a scheme of this embryo at the stage where all of this happens. This is, the way we dissect this region. It's roughly cut in those two axes. Then the somite region and the notochord is actually isolated from this unlag of the aorta, gonads, and mesonephros. And this is how it looks like. This is a scan of this region, and this is actually how it looks like once we isolate and once we dissect them. This is how it looks like. By using a mouse that has a GFP knocked in the FLIC1 locus, those are the images that you get. Now, FLIC1 is a receptor for VEGF-A, and it's expressed on all endothelial cells, at least during mouse embryonic development. Interestingly, GFP is also expressed on what we call the intra-aortic hematopoietic clusters, or YACs, seen here, expressing GFP, expressing markers of hematopoietic stem cells, such as c-KIT, CD41, and CD34. So in the mouse, which expresses GFP in all endothelial cells, the very first hematopoietic progenitors are actually expressing GFP as well. This is true for the aorta, as I told you. So in this AGM region, the main region that contains those early progenitors are the aorta, but you find them also in the omphallomesenteric and vitelline artery, showed here. So now, the omphallomesenteric and vitelline artery goes through the placenta, through the yolk sac. So I would like to offer an alternative explanation to the fact that people found a lot of this very early hematopoietic progenitor activity in the yolk sac and in the placenta. I think this could be explained by the vessels that, penetrate both the placenta and the yolk sac, and where this activity is actually heavily seen. So this is a general property of the blood vessels, of major arteries in the embryo. And I'd just like to show you a flow cytometry view of this same region that shows that all the cells that express GFP, either high or lower levels, can be subdivided according to the expression of CD45 that express on all hematopoietic cells, c-KIT, and in a double negative population. All the multipotent progenitors, and in fact the long-term reconstituting cells, are in the c-KIT, CD45 low fraction. They express very high levels of GFP, and in fact the higher levels of GFP, the better they are in cloning capacity. Now, the CD45 positive cells at this particular stage, they do not express GFP or very low levels. They do not contain multipotent progenitors. They are mostly myeloid cells coming out of the yolk sac. The double negative fraction actually contains mostly very highly GFP positive cells. Those cells also express CD34. We can actually grow them in colonies if you add in VEGF. Those are typical morphology of endothelial cells. So this is just to tell you that very high levels of GFP are expressed both in a KIT positive and in a KIT negative fraction, and they correspond to endothelial cells, and to what I'm going to show you later are the newly generating hematopoietic progenitors. I should actually point out that at this the same stages, we don't find cells with a similar phenotype, neither in the yolk sac, not in what will then become the placenta, okay? Whatever that means. All right, so what I'm going to show you now is that, in fact, very highly expressing GFP cells, as I told you, can be subdivided into KIT negative and KIT positive. Those are the first hematopoietic progenitors. Those are the endothelial cells. Now, if you take them at this stage and you sort them out and ask a very simple question, although they express very high levels of GFP, are those guys also expressing FLIC at that stage? And the answer is no. Only the endothelial cells are expressing very high levels of FLIC. The KIT-positive cells are not expressing FLIC1. And they, in fact, do not express GFP either, messenger RNA for GFP. So basically what we are seeing is, as those cells are generated, they express very high levels of FLIC and GFP, and immediately they shut it down as they differentiate into the hematopoietic or commit to the hematopoietic lineage. And what we're seeing afterwards is the dilution of GFP by either cell division or decay of the protein simply. GFP has a higher half-life. And if you look at the fetal liver at day 12, you find the endothelial cells that express very high levels of GFP, and then another population that express lower levels of GFP. Those cells are actually all they have all markers of hematopoietic stem cells in the fetal liver, meaning they are KIT high, SCA positive, CD150 positive, and CD48 negative. And they have, of course, a little less GFP than the ones that you find at day 10, because in the meantime they divided already, and probably the protein decayed. So the conclusion we took out of this was that GFP expression in c-KIT positive cells could be used to track newly generating hematopoietic progenitors. And so we decided to go and find out where those cells were. So first of all, what I'd like to tell you is that we can start to detect those cells by the stage of 25 somites. That's pretty early, like day 9.5, early day 10. They increase, they have a peak at like day 10.5, and then they decrease by day 12.5. You find a few cells in this particular region, but not that many. Now, if you look at GFP expression, you see that already by the 11.5, all those progenitors that you see here have lower levels of GFP than at day 10.5, where you actually find the same number as the ones detected by a functional in vitro assay, multipotency. So in fact, what we conclude is that the production is done between day 9.5 and day 10, 10.5, only in this time window, and then a few cells stay around. A few cells probably go into the fetal liver, and I'm going to tell you that we have good evidence for that. It's not particularly surprising, though. And that after day 10.5, there is no longer production. The cells stay around and, you know, lose GFP either by cell division or by cell division or by decay of the protein, and so this places the production in a very short time window between day 9.5 and 10.5. And this is just to support those claims. This is the levels of GFP compared to the ones in the endothelial compartment shown here as day 0. This is sort of, you know, rounds of dilution of GFP. By day 11.5 in this particular region, most cells have lower levels of GFP than those ones. There's basically no cells that express the same as endothelial cells. Okay, so the time window of production is very short, and where do we find them now? We find them now back in the fetal liver. Remember that at 11.5, in the HGM, you could no longer find very highly expressing GFP hematopoietic progenitors. They are all in the fetal liver. You find them also at day 10. You find them at day 11. And of course, by day 14, those cells diluted the GFP. Okay, so we find those cells at very early stages. However, we know that only after the 11.5, in fact, fetal liver can be long-term reconstituting in lethally irradiated recipient animals. So this is basically the type of experiments we do. Long-term reconstitution, we take hematopoietic progenitors of a CD45 allotypic congenic strain to the recipient mice. We inject the cells, and then we follow up the bone marrow 6 months later. This is the general test for long-term reconstitution. This is the result of when you take fetal liver cells and you inject them in such a wild-type mouse. This is, donor-derived cells detected by the CD45.1 marker. Now, when you do the same game with day 10 AGM cells, you actually find no reconstitution whatsoever. However, if instead of using a wild-type recipient mouse, we use a reg-MSC knockout mouse, now you see back, a little more modest, but still reconstitution in these guys. So in fact, reconstitution of reg-gamma versus wild-type became our assay system to distinguish between the newly generated, what we call now immature hematopoietic stem cells, and we know sort of why this is happening, as compared to, mature adult-type hematopoietic stem cells. So we decided to go into the fetal liver at very early stages. show that they do not, in fact, reconstitute wild-type animals. This is new. This is known for a long time. Now, if we inject those cells into reg gammas, now we find reconstitution, and if we take those fetal livers, incubate them for 3 to 4 days in an organ culture so that they further mature, and we inject them now in wild-type mice, we get reconstitution. So this is sort of like to show you that the first hematopoietic progenitors that colonize the yolk sac are actually what we call immature hematopoietic stem cells, but that those cells further differentiate and mature in this organ. And this is sort of the summary what I was trying to tell you. There is a first generation of cells in the yolk sac. We don't think that those guys will make hematopoietic stem cells. A second generation, we think those guys will make hematopoietic stem cells. Now in the fetal liver, those guys are going to expand and differentiate, colonizing the bone marrow and the spleen, and here they don't expand, but they just self-renewal. So this led us to think that fetal liver was probably a particular environment where actually hematopoietic stem cells expand, and it's the first site where they go. So then we decided to look into the hematopoietic stem cell compartment in the fetal liver and ask sort of very simple questions. Why do they express, in addition to the conventional markers already known of? And we stumble upon LFA1. LFA1 is an addition molecule that binds ICAM1 or CD54. And when we look at dividing hematopoietic stem cells, here marked by Ki67, we actually notice that the cells that expressed high levels of Ki67, meaning the ones that were more actively dividing, are also the ones that express higher levels of LFA1. So there was sort of a correlation between LFA1 and Ki67. So, and this is just a calculation of the total numbers of HSCs in a mouse, and how many of them are Ki67 positive, and how many of them are LFA1 positive, and there are none in the adult bone marrow. So we decided to go into the fetal liver and ask a very simple question: can we find cells that express ICAM1? Are those the partners of the hematopoietic stem cells? And so we took, again, this flick GFP mouse because it allowed us to distinguish endothelial cells from all the rest. This is the CD45 STER109 negative fraction, so there are no hematopoietic cells, no red cells here. And we subdivided them into 3 fractions. So we find the subset that is indeed ICAM positive, endothelial cells, and another subset that is GFP negative, and a double negative fraction. We divided that, and we call them fraction A, fraction B, and fraction C. What other markers are there expressed in those guys? So this fraction A and fraction B, meaning both ICAM positive, GFP positive, and GFP negative, they share some markers like VCAM1, CD54, as I told you before. There are markers that are specific for endothelial cells, like CD31, CD34, AA4.1, and there are markers that are specific for this fraction A, as we call them, like CD133. So, you know, those are probably not endothelial cells by this stage, and we decided to go and test them. And the test we started by doing was a test for messenger RNA for cytokines, because any cell that is supporting hematopoiesis in the fetal liver will probably have to produce some cytokines. And we knew that from, many cytokine knockout mice that we had dealt with in the past. So we all compare, this is a qRT-PCR, compared to the OP9 stromal cells. This is a bone marrow-derived stromal cells that we usually use in the lab to differentiate hematopoietic progenitors. So fraction A expresses most of the IL-7 that's produced, thrombopoietin, KIT ligand, SDF1, CXLT1, VEGF, you name it. I mean, all cytokines we looked at so far are mostly expressed in this fraction A. Endothelial cells also express some KIT ligand that is known they do, but much less than this fraction A. And, you know, by and large, it's much better than the OP9 stromal cells for anything. Okay, so the next thing we did was, can we grow those cells in any possible way? Can we expand them? So we took fraction A, fraction B, and fraction C again. So this comprises all the hematopoietic negative compartment of the fetal liver, and we tried different ways to grow them. So the fraction C grew in 20% FCS normal medium and gave rise to cells with this look. They look like fibroblasts. They look like any stromal cells. They look like mesenchymal cells, basically. Fraction B, as predicted, grew this way, only with VEGF-A added to the culture medium. Those are typical endothelial cells. Fraction A, which is the one that produces the cytokines, grows like this in colonies of this look. They only grow when you add in insulin, IGF-2, and EGF. And they looked by morphology to be, you know, epithelial cells, and people tell me those look like hepatocytes. All right. So can those cells do something? So now that we knew how to grow them in a very short term, and this is like not more than 1 week short term, we actually attached them to the bottom of a well and put on top of them pro-B cells from the bone marrow and asked, can we grow those guys? I mean, this is all exogenous cytokine-free. And this is the number of pre-B cells at the end of 5 to 7 days, and the number of B cells that we get out of those cultures compared to OP9 cells, compared to fraction C. And we actually get a lot more of those pro-B cells than in OP9 cells, and a lot more of B cells. And this is, of course, consistent with the fact that those cells produce interleukin-7, that is a strict requirement of bone marrow pro-B cells to develop. So can they do anything to hematopoietic stem cells? Actually, they can sort of expand them by counting them by phenotype. So if we take LSKs at day 0 that are CD48 negative, CD150 positive, this is the starting population at the end of 3 days. We have this population like this, which with the numbers recovered per input. So they sort of like doubled in 2 to 3 days in culture. And of course, the next question was, are those long-term reconstituting cells? So in fact, what we did is we isolate those cells by FACS sorting. We put fetal liver LSKs on top of them. We waited for a few days, and we injected them in lethally radiated recipients in a competitive long-term reconstitution assay. We analyzed the blood at day 40, 19, up to 210 days. And this is the result. This is myeloid reconstitution at, you know, those different time points ended up in 210 days. LSKs grown on top of fraction A. Fraction B, endothelial cells. Fraction C, medium alone. So you only get long-term reconstitution in the cells that were incubated on top of fraction A. This is the number of B lineage cells that you find in the spleens of those animals. And of course, this is highly significant. And this is the numbers of not only LSKs. We actually went in the bone marrow of these guys and we looked at CD48 negative, CD150 LSKs as well as LSKs. You know, they are highly there. So, you know, those guys basically sustained hematopoietic stem cells. So, because they looked like hepatocytes, by morphology, we actually decided to look whether they also expressed anything that hepatocytes expressed. And we analyzed them for AFP, alpha-fetoprotein, albumin, HNF4, it's hepatocyte nuclear factor 4, and FLIC1, which of course we knew was expressed in the endothelial cells. And you know, those guys are the only ones that really express AFP. As compared to everything else. This is compared to adult liver cells. They express albumin, of course, adult liver a lot more. They express HNF4, more or less the same thing as adult liver. And of course, they don't express FLIC1. And in a multiplex single-cell PCR analysis, we actually show that those cells, the same cells that express HNF4, also express KIT ligand TPO and IL-7. You know, most of them actually express HNF4, KIT ligand TPO. IL-7 is pretty rare altogether in fetal liver. And KIT ligand, HNF4, of course, you don't find HNF4 in OP9 stromal cells as comparison. So those are the same cells, by and large. That's the conclusion. Where are they? They are in the fetal liver, very rare at day 11, more frequent at day 13, peak at day 15, start to go down at E17, not detectable in newborn mice. So this is a transient population, not only by phenotype, but also by cytokine messenger RNA expression. They are not there. The TPO expressing cells are not there in the newborn compared to the E13. IL-7 producers are not there. KIT ligand are not there. Albumin, they are there. The fraction, you know, some cells around here. Alpha-fetoprotein, they do express it much lower than the newborn, of course, because much lower than day 13 because AFP is not expressed. Those cells are not cycling. They are the only cells in the fetal liver that are not cycling. This is a Ki-67 analysis of fraction C, highly cycling. Those are the endothelial cells. You know, you have some cycling, some non-cycling. These guys are mostly Ki-67 negative. And, you know, if you go and look at intracellular HNF4 together with Ki-67, those guys are not high for Ki-67. So they seem to be post-mitotic. So just to finish up, I'd like to tell you that we also find them in human fetal liver. So we teamed up with Helen Strick-Marchand and Jim. They actually get human fetal liver to make humanized mice, and I use the fraction that they don't want. This is just to show you, you usually isolate those cells by a fical gradient. You have a fical ring and a fical bottom. CD54 positive cells that actually also express, that do not express CD34 but express HEPCAM are found mostly in the fical bottom of these fetal liver preparations. Most of those cells are CD34 negative, so they express CD54, they are EpCAM positive, they are mostly c-KIT negative. There's exactly the same phenotype as the cells we isolate from the mouse fetal liver. And interestingly enough, they are the only cells that also express HNF4, just like in the mouse fetal liver. So we have cells with the same phenotype. We are now analyzing their capacities to support hematopoietic progenitors from human fetal liver and then try to inject them into humanized mice to see if they really can maintain hematopoietic progenitors also in the human case. So I'm going to finish up here. These are the conclusions. In fetal liver, hematopoietic cytokines are produced by a transient population that express CD54. They exist in the mouse and in human fetal liver. They seem to belong to the hepatocytic lineage, but they are transient population of cells. They seem to sustain hematopoiesis, but also HSC survival and expansion. So this is the last slide. Those are the collaborators that did that. Helen, Isabel for a long time, Alexander. And of course, this is the pastor where I started all of this in actually Philippe's lab. That was great and gave me the chance actually to develop all this on my own. So I'd like to thank him again and all of you for listening to me. Thank you very much.